Okay. I think he's going to retire or already retire.
Yeah, he's. I heard he's. Andrew knows, but since he may be retiring soon, well.
So have not retired yet.
Oh, what happened? It shows. So not showing what happened. Okay. Live shows this.
Okay, Chiru, ready?
Yeah, sure. Okay.
Okay, welcome everyone again. And Chiru today is talking about so many kinds of amacrine cells and from Sain's lab. Okay, please.
Yeah, sure. Oh, okay. And today I'm talking about a paper from Joshua San's lab, and they use a method to identify over 60 amacrine cell types. So the result is that the find types will be increased to 63. So the method they're using is the single cell RNA sequence. So in pre-study, they think that they can like analyze all of the cells at the same time, but since the cell numbers of photoreceptors are so much, so in the previous study, find out that those amacrine cells are endosampled. So in this study, they use a selective depletion. So they isolate the retina, and this retina is from CHX10 and GFP transgenic mice. And this mice line have GFP on the bipolar cells and the mular cell. And then they also stand those dissociated cells with anti-mouse CD133 that's labeled COM, and the CD73 that's labeled the rod photoreceptor. Then they use the facts to remove all the photoreceptors and bipolar cell and the mular cells, and only there's a ganglion cell and the amacrine cells are left. So they isolate these cells.
Sorry, Chiru.
Yeah, yes.
So that's a VSX to label all the bipolar cells?
CHX10, I think it's a lot.
Majority.
I think it's a lot, yeah. Mm-hmm.
Okay.
And they only isolate those cells that's the canceling blue marker, and that's the live cell. Then put in this kind of device that the cells and enzyme from one way and those beads from another way, then isolate a cell and enzyme and the beads in a droplet that's an outside isolate by an oil. Then within each droplet, the cells was lysed and the MI are released. Then MIs are binding to those beads. So they collect the beads, then do the sequencing and analyze the transcript profile of those isolated cells. In addition, they also confirmed with fluorescent in situ hybridizations to match the gene expression profile with the cell morphology. In the letter, they will mention that there are two types. They have very similar expression gene profile, but the cell morphology is dramatically different. So here is a simple description of the job sequence, that the cells are isolated within the job, and that's resynthesized the cDNA library of those RNA transcript. And the beat have like the arms, and there's a three part. One is the PCR, then a PCR handle. This is all same from all of the beats. And the next sequence is the cell barcode. And for each cell, for one bit, all the cell barcode are the same. And the third sequence is a unique molecular identifier. And this sequence was different between these this sequence. So in this way, to analyze if the because they put all this bit together to do the sequencing. So all the sequence that with the same cell barcode means that they're from the same cell. So it's like a cell identifier. And those cDNAs with the same sequence of this UMI mean some, for example, this one and this one have the same, like a binding with the same mRNA. So this means that like every 8 MI, and 2 of them are the same MI. So this can evaluate the transcription percentage of all MIs. So this is they also used several transgenic mice for the CHX10 GFP, create GFP mice, they to isolate, to get rid of the bipolar cells. So they use these lines. And other lines like this SLC mice and PNK mice, those lines are usually think that there's like express CRE in amacrine cells. It's like amacrine cell transgenic mice lines. And the data sampling, after data sampling, they do the plot. They using this UMAP method, and this is a non-linear method. And this distribution is using a Riemann manifold. So it's like they put all the data is not on a 2D flat, like XY on this, but they put all the data on a curvature, like on the surface of a ball, and then do a 2D dimensionalization, and then get this method. So this is their result. They find that the Amacrine cells have 63 clusters. This is their result from this paper.
Excellent.
And they also confirmed with cell morphology. They use the traditional antibody markers, and also they use some dependent GFP expression. That's like the Cree animal then inject with AB GFP, and the GFP expression is dependent on the Cree, and they reduce those virus concentration to very low. Then in this way, they can just for the whole retina, they can just can separate those cells that are Cree positive, then to make the cell's morphology more clear to be seen. So the result is shown in this table. And 10 years ago, the total number is about 65. And the ganglion cell is 20, bipolar cell 12, and amacrine cell 30. And now after 10 years and nowadays, the bipolar cell types increased to 15, and the ganglion cell type also doubled to 46, and the amacrine cells is also doubled to 63 types, and the total neuron in the mouse retinas cell types increased to 128 different types. So in this study, they studied amacrine cells. So they want to confirm that all those data are exclusive is amacrine cell. So in previous slide, they described that the way like how to the selective depletion that those the GFP get rid of those bipolar cells and photoreceptors. So they test the genes in those cells. Let's do the way, find that those patterns are not the amacrine cells gene patterns, but fall down to other types. So and also, okay, so this is another description to say that from like the 20 years ago, the mass dance result show that like most of the cells are rod and comb. The amacrine cells cell total number is like around 7 to 10% of the total cell. So, but the cell types of amacrine cells are huge. So their result show, so this is the figure 1F, and it shows that so most of the amacrine cells are either glycinergic or GABAergic, but they still find some types that have both GABAergic and glycinergic. And also they also find 4 types, and the total is 6% of the amacrine cells is non-GABAergic and non-glycinergic. So they release other transmitters or neural
Actually, can I ask a question?
Yeah,
So
sure.
it's
Yes.
a little bit get confused. So this Maserand data 20 years ago and Jan's current data, it's really different. And so ganglion cell, is that a population number of cells?
This the number, and this is cell type. This is not the cell density. It's not a cell number.
Type number? Well, I don't know. So that I thought that amacrine cells is like amacrine cells and ganglion cells is about similar number, like a type number or population number 50% each. That's what I thought, but it's this Jan's result. It's amacrine cells much more than ganglions. That I get confused.
Yeah, I think it shows yes. So okay, their data shows the cell types. Amacrine cells this one, ganglion cells this one. So 63, 46. Yeah, it's I think this one. This figure D is from there. I think
so ganglion cell is lower than bipolar.
30.
Talking about what you're talking about.
So the cell fracture of cell in each cell class. Fracture of cells. Fracture of the amacrine cells. Fracture cell. Okay. Well, yeah. But this one is the cell density, that's the total cell number. And this one is the cell type. But although I think this image, then let's see this. So these are also the cell number. Maybe this from cell number. Oh no. This only This is only amacrine cells. It's not the all of the cells. So may I move on? And so they also compared their result from 2015 Moscow's the cell type study, and they claim that the cell types are contained all the types that are described in 2015, but 2015's result like put so they separate more, and they didn't miss anything. That's what they claim. Like for example, the 12 and the 15, these two type. In the 2015 study, they think that it's the same type, it's the same cluster. But in this study, they think that they separate them to be two different clusters. And their studies are from P19, that's like a 3 weeks old animal. And they also compared with the P56. They also compared the P6, the adult data set. The adult data set are also from this 2015 study. And they also find out that their results are contained and the adult data set, the P56, and they didn't miss out. So in this way, they claim that their studies didn't miss anything and find everything and separate the different types nicely. So they put all the 63 types of cluster and with a gene, with different genes. In this plot, they can find out that each cluster can have a very specific like marker genes for each type. And this they show that, and this I think it's a VG3 amacrine cell, and it expresses this gene CAR3. So it stands with the CAR3 and it's co-localized with the VG3 amacrine cells. So the VG3 amacrine cells is in this. So it's in this. It's the cluster 35 type. That's the VG3 amacrine cells. And this image is from Cree mice that was injected with Cree-dependent AAV-GFP, and then stand. So these cell types express GFP in this specific type of amacrine cell, and that's cluster 23, and it's co-localized with this gene CD01408. And and the VSHT is the chat band. So later they will use a lot of images with this VSHT. That means that it's a chat band. So for very famous.
Can you go to the previous slides? I want to take a look at that one. Okay.
Yeah, sure.
The previous one.
Oh, sure.
Okay, so this diagram means each particular American cell going to express in most cases is a dominant one type of the gene. Because that line indicate, right?
Well, it's like all like for each cluster, each type, all these expression gene patterns will determine that. Because but if they put all the data in this way, it seems like um
So for example, this type of 5 that's is dominant express this particular gene. Is that right? Because the dot is larger. So
yeah,
of course,
that's right.
in some American cell, maybe let's say, yeah. So in general, like each American cell is expressed one dominant gene. Is that in general, right? So that's line we indicate here. Okay. I don't want to make that.
Yes. Each circle, if it's large, so that means that in this specific type, most of the cells or the more cells that express this gene. And if this dot is very very dark, that means that in each cells that the expression level is very high. So some dots is small, so it means that there's a less percentage of this cell cluster express this cell. But for those cells that express this gene, the expression level are very high. So there are two, each circle have two, the darker there's a more gene, and there's a larger means more cells. And this shows several very famous amacrine cells like starburst amacrine cell, A2, A17, and also show this use monoamine and amacrine cells. Like compared in this image, they compared this cluster and with the adult data set. It's like this is from like a P19 cell, P19 animal, and this is P56 animals, and their expression level are quite similar. So make no very big difference between like a P19 and the P56. But that's for sure that's like newborn, like a P0 and to P19, that would be a huge difference. That's true, but yeah, 3 weeks are quite similar to the adults. But there's also some specific example, like this cluster 45, and they have a very much higher expression. It's not higher expression, it's more cells, right? More cells express those dopamine synthesis enzyme in adults, but in younger there's less cells express this one. So here it shows a developmental difference. But generally other cell types is like P19, P56 are quite similar. So we know that most of our amacrine cells, the transmitter they're using is either GABA or glycine. And they study and they confirm this, and that's true. For GABA, like a GABAergic marker, the GAD1 and GAD2. So that's a synthesizer, right? Transmitter synthesizer enzyme. Yeah, GABA synthesizer enzyme. Yeah, many types produce GABA, and some other groups that produce glycine. And this is ACLC33A1 gene is like a amacrine cell marker. But they also find like three cluster, like a 16, 53, 62 or 62. And these three types, they both use GABAergic and glycinergic, GABA glycine, GABA glycine, and it's and this and its morphology staining show that these cells have both glycinergic and GABAergic and colocalized within the same cells. So there are 3 clusters are in this type. There are also some amacrine cells that they have this vascular glutamate. I missed something. Vascular glutamate transporter, gluten 1, and it's like a marker for the VG1 amacrine cell, and gluten 3 is a marker for the VG3 amacrine cells. And these cells, when you see that this VG1 amacrine cells marker is like they are positive of the AP2. AP2 is actually a amacrine cell marker, but it's a VSX2 negative. So this is VG1 amacrine cells, and here it's this one. Let me see. And it's a C56. So this image from this cluster of amacrine cells, and several some amacrine cells also used other neurotransmitters like starburst amacrine cell, and here is the cluster 17. And it's cholinergic, and it's very interesting that it's the only cholinergic amacrine cells. Other cells are it's.
So I can ask you a little bit question. So that's like this one also glutamate, vascular glutamate gene. So it's a big bubble. I understand 17 expressed chat, really large bubble, but even others also show the kind of tiny dots there. So they are.
Yes.
How do you think about this? What is a kind of cutoff? Kind of like this is meaningful as a chat cell or as a where's the cutoff like? So others are also cholinergic potentially, like a 24, number 24, almost like a later phase. That's also kind of almost middle size of the expression level. Yeah, is that considered to be cholinergic, or it's you don't have to worry about that? That's my question.
Yeah, I think in this paper the authors they describe they discuss about that. They say that yeah, maybe there's not so much, but we cannot say that because they have this, but although it's weak, but maybe they're also involved in the synapse transmission. They use one sentence to say that. But starburst amacrine cells are very like very obvious, much so very different from other types and quite clean.
But like a 24, 10 from like almost right side, it's
24.
Yeah, 24 shows like I don't know what
here,
kind
right?
of,
24.
Yeah, 24.
24. Oh yeah.
Yeah, that one. It's like a chat is kind of there.
This chart is small. The chart is small.
Of course, smaller than the 17.
Yeah, it's smaller. Yeah, this
still
this
there.
SLL is a amacrine mark. I don't know. It's so strange. Previous every cell type have very large this circle.
So the question is, so I mean, so this technically, even how much technically contaminate, that's another issue. I mean, so they have to put a threshold, like the watch level is reliable, or the watch level is just due to contamination, or just because it's really the low gene expression. So that's why I'm not sure here.
Yeah. I think that's another issue.
So this drop-seq, like a contamination can happen, like maybe underuse here.
I think because they are using low speed and they do the RT-PCR all the beads together, I think that's possible because they just mix all of them together. Yeah, I think that contamination is quite highly possible. Yes.
I also think
Yeah.
part of it's just it's a little messy because there's so much amplification that you get some like low level messiness.
Yeah. Yes. Yeah. Probably the RT-PCR is so sensitive. And there are also some cluster of cells that use as the monoamine synthesized enzyme. They also find some cluster that they can produce the nitric oxide and the carbon oxide. Oh, it seems like all of the cells can generate carbon oxide. So it's like this gas is a traditional.
Carbon oxide.
Yeah, the carbon oxide and everyone.
Carbon oxide. CO.
Why?
CO. That pollution.
Cool.
Very interesting.
And yes, and Amacrine cells also use a lot of neuropeptide because they have a gene for a neuropeptide precursor. And there's several retinal calcitonin and cocaine, amphetamine regulate transcripts, ganglion, like growth hormone. And yes, Amacrine cell produce a lot of neuropeptide. And this, I think that this they cluster like and this is analyzed this the PENK Amacrine cell because there's a very specific transgenic mice that's the PENK CRE or PNK GFP. But in this study, from one transgenic PNK Amacrine cells, but actually there's 3 types of Amacrine cells are involved, like class 35, it has a very strong expression of the PPP1R17 and this gene, and cluster 59 and 63 have very low of this, but also low of this seems that it looks like not colocalized, and cluster 63 have very high expression of the SST or somatostatin. So I think that kind of like reinvestigate those transgenic mice or knocking mice to see that before maybe seeing that there's one type, but actually maybe there's several types within one transgenic mice, all labeled with the same with the GFP. So here is a summary said that all Amacrine cells types express a neuropeptide. And in this study, they also described the non-glycinergic, non-GABA Amacrine cells. So in previous study, it's only found one type. I think it's the cluster 10, the number 2. But in this study, they claim that they find like 4 different clusters that do not use GABA, do not use glycine, use as their transmitters and therefore types. And so for the first one, cluster 24, and they stand with the PPRP, and that's a very strong expression of the PPRP, and not very high expression of the NFI6, and this non-glycinergic and non-GABA Amacrine cells so interesting because in previous study find that these Amacrine cells are able to re-enter the cell circle and generate new neurons and glia. So I think that's why maybe they're quite focused and study on this type, non-glycinergic, GABAergic. And the cluster 10 is the second type of NG. And so we can see here that there's when with co-labeled with glycine transporter and the GAD, so they are negative, so they're non-GABAergic, non-glycinergic, but they have some other gene markers like in the second 2. It shows also very high, so it's EBF3. Let's see. Okay, have expression EBF3. And we also have not very high expression of the PPRP. So although the circle is small and quite medium color, but immunostaining is so weak. So I think probably this method of the RNA sequence is more sensitive than the immunostaining. And it also show the morphology of this this type 2 NG. It show that the terminal is like this. And we can see that the expression pattern actually is quite similar between this type 2 and the type 3. We can see everything. Yeah, I think it's quite similar. Although there's some expression level difference, but if I like yes or no, it's similar. But the type 3, the morphology is quite different from the type 2. And if we can see this image, this is the type 3. And compared to this, I think it's quite different. So although their expression pattern profiles are similar comparing to other types, but their morphologies are quite different. And this one is the cluster 36, is the number 6 of the NG type, and confirm that it's not GABAergic, not glycinergic, but it's a high in this LHX9, high in LHX9 here shows high, and the cell morphology is like this. It's on between the chat band. So to summary, so this year bipolar cell number to 15 type, Amacrine cell to 63 type, and ganglion cells to 46 types. This image is from this research, and they also compared these 3 different types of the genes that I think most of them are like receptors, and so generally there's still a large difference. For example, for the GABRA1, GABB2, and Amacrine cells have low expression, but it's very high in ganglion cells, like DRD4. DRD4, I think is dopamine receptor. I want to confirm that DRD2. Oh, okay, it's dopamine receptor, and bipolar cells and ganglion cells have very high expression of this GABRA, and this GABI. It's GABA-A receptor subunit alpha 1A. So but it's low in Amacrine cells. But here, I find it interesting that they show that the GIK4, the inotropic, the kinetic type, probably is the most difference between off-type bipolar cell and on-type bipolar cells. So this is a table show that the result of Amacrine cells in 63. The total number is 124. And they also Amacrine cells also use a lot of neuropeptide and a neurotransmitter. So this is a summary of all the neurotransmitter and neuropeptide that you see. So another another like let's separate a conclusion of this paper like correlated with the transcription factor with the functional, like the transcription factor of the MACE2 and is mostly is a GABAergic, and the transfactor of the TCF4 and mostly is a glycinergic. So it's kind of correlated with the transcriptional factor then with their future functional. And the cluster 6 is have both cluster 6, let's see here, cluster 6. Well, but not very strong. Yes, both positive of TCF6 and MEIS2. And then we go to take a look at like the 1024, like, oh, okay, yeah. So here is a summary of the previous that the glycinergic that it's positive in a TCF4, and the GABAergic are positive of the MEIS2. So probably like MEIS2 transgenic mice probably can label all of the GABAergic Amacrine cells. So this is a table show that some Amacrine cells are glycinergic, some are GABAergic. And there's a description of the different clusters. So the overall summary and different cell types. Some cells, because they have like the markers of transgenic animals, so we can label it like A2. A2 is here in this cluster, and like a VIP F3 cluster share the same marker, and also some like NG cell types. So they compared with the previous studies, because other previous studies have markers, but the most abundant of Amacrine cells find in this study it's like not subject to previous study, like cluster 1, 2, 5. As the no markers, it's not very specific study, but their cell numbers are pretty high. They also discussed about the technically, so maybe some cells still lose because they are very fragile, so maybe loose and damaged. Because before this step, I should have live cells and intact cells. But they can claim that they think they got like 99.9% of correctness. So I think that less than 0.1% of Amacrine cells are missed in their study. So I think they're quite covered everyone and everything. And they also described here, Here they also are talking about on and off starburst amacrine cells. Then when in the newborn, starburst amacrine cells gene expression profile is quite different between on and off, but after like 3 weeks at the P19, and it's quite similar. So it's the same type. So for this functional study to analyze the expression pattern, we're not separate the starburst amacrine cells after P19. And although the total find out that there are 63 amacrine cell types, but most of them, like 56 types, are either glycinergic or GABAergic. Glycinergic, that's a 30 type. GABAergic have 43 type. Then total is 56 types of amacrine cells out of total 63 types. But as we still find some spots like this one, like this circle is point that some, it seems these cells are different. It's been separate in the analyzed study, but they still cannot tell it's a separate cluster or something else. So there's still some very mixture like here. So I think maybe their method can still improved. GABAergic outward field. Oh, okay. Then they also discussed about the morphology difference is quite separate from the gene expression pattern, but generally say GABAergic amacrine cells are wide field and glycinergic amacrine cells are narrow field. So maybe in the future they can go to the expression profile of like other pathological animal model, like RD mice and other lines. Yeah, that's it. That's it.
Thank you. It's a really informative paper, and you presented very well. It's very informative. Any questions to Chiru?
Yeah.
Well, it looks like we have a lot of work to do to identify the physiology of all these amacrine cells. It will take another 100 or 200 years.
So are they all on Amacrine or off Amacrine, or there's no such a marker available?
Yeah, some like yeah, it's here in this figure because some profile very close, basically, and the only difference like a couple gene. Okay, so I don't know how accurate so separate some into different type. That's another question. If you go
physiology, they are same, probably. Or even morphology, they probably same too.
Yeah, wider field and narrow field exist.
And also, I think it would be nice to correlate the morphological property, right, in the whole mount, because if the cell type really have a unique type, then suppose it should be have kind of a unique pattern, right, across the. That would be another very important indicator. Yeah, okay, anyway, so this makes things more complicated. but at least right now, so the stable Amacrine cells still have a single type, right?
Yes.
Based on this paper.
Yes. I'm glad. How about TH cell?
Single type.
Single type. How about TH cell? Just this eight.
TH cell.
I saw a couple of the TH.
This TH here, cluster 25.
About like a number 3, like right side, or
Number 3.
number 45, it's a little bit
hmm.
heavier dots, number 3.
Yeah, but maybe they're different in other ways. Yeah, in other genes. Yeah.
Wow. I'm glad it's still like same big picture, like either half of the GABAergic, half of the glycinergic. Majority is either GABAergic, glycinergic. I'm glad to hear that.
Yeah. Yeah, I think we can say that. Yes.
Do they have information about glutamate receptor?
Yes, I think this one is the
This is
for
vesicular glutamate.
for the ganglion cell marker. This one shows very low expression level. So yeah, but like a type like a C50.
Oh, no. I'm talking about glutamate receptors. So
Glutamate
they all get the information from bipolars?
Oh, let's check which one. This is not peptide. Yeah, I think I see one. Oh, maybe this one, glycine glutamate receptor, intrinsic receptor. So you mean
AMPA NMDA.
top. So it's glutamate. So most of it on the top. Yeah.
Oh, all of them have. Okay, I see. Very strong. Also NMDA also strong too. Wow.
Let's see. Glutamate, yes, GRI3 is from this GRI3A here. The alpha subunit.
GABRA. Yep. I see. Interesting.
Okay. Then
Any other comments, Cecilia?
me? No.
Okay. Seems to be everyone understand. It's very understandable. Okay, thank you very much.
Thank you.
Yeah.
Thank you.
See you next
Okay,
month,
see you.
everyone.
See you.